Our next speaker is Michael Snyder. As a pioneer of precision medicine, Dr. Michael Snyder has invented many technologies enabling the 21st century of healthcare, including system biology, RNA sequencing, and protein chip. Dr. Snyder has initiated the big data approach to healthcare through his work using omics to detect early-stage disease, including wearables to detect infectious diseases like COVID-19, and at-home microsampling to measure hundreds of molecules from a single drop of blood. He's the first researcher to gather petabytes of data on individual, which is 1,000,000 trillion times more data than the average clinician collects. He has published over 800 papers and is one of the most cited scientists. In terms of commercial success, Dr. Snyder has co-founded 17 companies, including 2 unicorns, with combined enterprise value of over $6 billion. Please help me welcome Dr. Michael Snyder.
Okay. All right, well, it's a real pleasure to be here. And Ranjan, I'm sure your running time is faster than mine, but I think my weightlifting is probably a little stronger. So we'll have to see what the right parameter for aging is. All right. Anyway, I'm going to talk about our work using big data to try to transform both healthcare and how we understand and deal with aging. And I think most of you would agree our healthcare system is broken. We really practice sick care rather than healthcare. And if you think about it, even when we practice healthcare, it's pretty antiquated. Typically, what you'll do is get in a car or some other mode of transport, always at a very inconvenient time. You then travel to a physician's office that looks pretty much the same as it's looked for the last 40 years. When you're there, they'll stick a needle in you and draw lots of blood and take from that blood, and the time you're there, actually very few measurements when you get right down to it. And from all those measurements, they'll treat you based on population averages, meaning how are you as on the average, but reality is you may be individually very different, and so we think you should be treating that differently. We actually want to transform every one of these steps, and many of you may know a number of years ago, one of those ways is to use big data. So we set up a project that we called Personal Omics Profiling, where the idea is to collect very, very deep data on people and do so longitudinally. So it's the smallest group of people we've been studying now for, well, one is me, over 13 and a half years. The rest are around 10. Some are dropping off. We're adding new ones. We're up to about 171 people that we've been probing for extended periods of time. We'll sequence their genome once, and then out of their blood and urine, we'll make as many molecular and omics measurements as possible, and the list is up there. We'll also look at their microbiome. This is on top of deep clinical testing, questionnaires, surveys. We track their lifestyle pretty carefully. And then we have some advanced tests like stress echocardiograms, glucose control measurements, because we're very interested in type 2 diabetes, because I'm type 2 diabetic. And then we also brought on wearables about 10 years ago when they were actually started as fitness trackers, and we realized they might be powerful health monitors. So one aspect, as I say, is to collect very, very deep data on people, and the other aspect is to sample them longitudinally while they're healthy. So we'll take samples roughly every 3 months while they're healthy, and then if an adverse event comes along, like a viral infection or some people have had heart attacks and things, we'll take additional samplings, usually 5 to 7 samplings. So you might say, why are we doing all this? We're trying to understand what it means to be healthy, not what it means to be sick. How does it differ between different people? What's the difference between you and the person sitting next to you? How does it change over time, relevant for aging? What happens at the earliest times of illnesses, like viral infections? And then certainly from the health management side, can we use these advanced technologies like genome sequencing, wearables, to better manage people's health? And I can tell you when we started this with genome sequencing, pretty much all physicians said that is a really bad idea to sequence healthy people because you're going to find things, you're going to turn everybody into hypochondriacs, it's going to cost millions of dollars, which hasn't happened. And in fact, now many physicians, not all, are warming up to this. I think they still hate wearables, but we're working on that too. All right, anyway, with regards to these advanced technologies question, just from the first 3.5 years, we had 49 major health discoveries from the first 109 people. So roughly half the people learned something pretty important about their health, all pre-symptomatically. So before symptoms had appeared. And it spanned a wide range of areas: hemoncology, cardiovascular disease, metabolic, and so on and so forth. And no one, some examples, if you look over at hemoncology, we caught some with early lymphoma, 2 people with precancers, MGUS, smoldering myeloma, 2 people had serious heart issues, One was picked up by genome sequencing, another by wearables. And that's another lesson. No one technology found this. Sometimes it was genome sequencing predicted risk. We had a bracket case. Sometimes it was the imaging, sometimes the molecular measurements. Usually a combination is what revealed these issues. Again, all pre-symptomatically. And so the way we like to think of this is that if your health is a 1,000-piece jigsaw puzzle, we're trying to have 500 or 600 pieces, a much more clear picture of your health, rather than 5 or 6 we would argue you do today. And so we think this has been fairly effective for finding, again, these things pre-symptomatically. Now, relevant to this conference, one of the things we've learned is you're tracking people over time is that you can see how they change. And we see shifts over time. There's about 600 molecules in our first analysis that change, many more as we've dived in deeper. And it turns out everyone, we interpret this as aging, everyone is aging differently. So for example, the person in the middle, their top biochemical pathway that's changing is their cardiac hypertrophic signaling pathway. And we later learned this person's stage 2 hypertensive, which sort of makes sense with that general idea. On the left, that person actually has metabolic and immune system changes over time, a pretty typical ager. We think you need as few as 5 measurements within 2 years. We have a pretty good idea how you're aging for most people. And so we actually call these ageotypes that we follow. At the time, we had 43 people with enough data to see how they were aging, and we've classified the 4 major ones we discovered there. I know there are more than 4. There are cardioagers and things like that that aren't up there. But if you look at the folks on the left, I don't know if we have a laser point. Anyway, the 4 folks on the left, they're aging in all 4 categories: kidney, liver, metabolic, and immune. Some people like the box on the right. That person's not much of a kidney ager, but is aging in other categories. The box all the way to the left, it's a kidney and metabolic ager, and the next box over is kidney. So again, people are aging in different fashions, and we can follow this. And there are clinical markers associated with this. If you want it to be actionable, you need appropriate markers. And so these are just some of the markers associated with the ageotypes, and people can act on these. I'll come back to this theme a little later. But each line is a different person. A dark bar means it's statistically significant. A very easy interpretable one is the one on the left. That's hemoglobin A1C, a marker of glucose. And you'll notice that 4 people have the group as a whole goes up over time with hemoglobin A1C. That's not a surprise. But 4 individuals do go up. Those are the dark bars that go up. But 4 other individuals go down. As I say, this is easy. It's easy to correlate why they went down. Some people went on diet, some people went exercised, and some people lost weight. And so you can make these correlations. And then there are other things that were kind of interesting, like the creatinine, a marker of kidney function. The group as a whole goes up, as you might expect, but 10 individuals went down, and 8 of the 10 are on statins. So we don't fully understand the mechanism of that, but it's kind of interesting to make these sorts of associations. Doesn't show causation, but I think it's very intriguing nonetheless that the folks on statins have actually reduced their creatinine. So more recently, we've added on more data sets. I won't have time to get into this. We've done a very deep lipidomics study, very understudied class of molecules. I won't have time to talk about that, but we can see very interesting patterns. Monty Moore here has actually worked on this. And then we've also added on 4 microbiomes, the gut, skin, tongue, and nasal. That turns out to be really interesting to see the correlative patterns going on there. But we took a look at all these, and instead of doing looking for linear changes, which is what most people do over time, we look for nonlinear changes. So there are a small number, and I mentioned some before. About 6% of our molecules show linear changes over time with aging. But if you do a nonlinear analysis, you actually find many, many more by looking at changes over time. And in fact, something like 81% of your molecules will shift over time during this period. And in fact, so the number of nonlinear molecules changing nonlinearly greatly exceeds those that are changing linearly. And then we can do all kinds of pattern analysis to look at this. Some of the biggest changes occur at the end. This is one type of analysis where we look for where the maximum change is occurring. And it turns out there is a peak towards the end as people hit their 60s. You see big shifts in molecular changes and microbial changes. Turns out there's a pretty big one around age 44 as well. So kind of intuitively, I think those of us who are past 44 know that these seem to make sense. And then if you actually dig into the molecules and pathways, you can see what's going on. Super complicated slide, but we can track the molecules that change at these different periods. So up there is the mid-40s peak, and the bottom is the peak that occurs after 60s, and you can again see the sorts of changes. And again, many of these make sense. For example, in the 60s, your immune system, I think we all know, shifts quite a bit. You can find others there as well. And yeah, lipid metabolism, alcohol seems to be one of the peaking ones in your 40s. You can interpret that how you like. The point is that you can actually see these molecular pathways shifting. And oxidative stress is one as well. And we would argue those are two key times, at least, when there are dramatic shifts going on biochemically in people. The other area we're pushing a lot is this area of doing remote monitoring. I think the idea that you go to a physician to follow wellness just seems very archaic. You know, people there are sick. Why, when you're healthy, do you want to go to a doctor to get yourself sick? So can't we do more of this at home? And so we've been pushing two approaches here. Many of you may know we push the wearables big time and something we call home microsampling, where we take drops of blood, mail it in, and we make lots of measurements. I know what that sounds like, but ours works. So anyway, on the wearables, there are thousands of devices out there. How many of you are wearing a smartwatch or ring? That's better. Okay. I would think this crowd, everybody would be wearing one. All right. Anyway, there are thousands of devices out there. I'm sure you know. I'm a big fan of them. I normally have 4 watches. One broke. I do have my ring and so on. These days, I also have a hearing aid, which I do use for hearing. But hearing aids are also sensors as well. They measure your interaction score. You heard about socialization. They also measure actually physiology. I'm not sure if whether they're more accurate or less accurate. We're measuring that now to the wrist device and such. But anyway, there's lots of devices out there. They're powerful because they follow you 24/7. And many of you may know, we actually discovered early on, we put these on our cohort, we found that you could pick up when people are getting infectious disease presymptomatically. And this is when I discovered I first got Lyme disease. I was actually flying to Norway, and it turns out my blood oxygen dropped abnormally low. My heart rate went abnormally high, and I later saw my skin temperature was off. This was all pre-symptomatically. Never came down to normal. I was on a plane when we landed, these elements were off, and I later did get symptoms off and on. I went to a physician. He drew blood. Sure enough, I had elevated monocytes, and he wanted to give me penicillin. I said, no, I need doxycycline. Got a little tense. He did give in and gave me doxycycline, which cleared it up. You take it 15 days. When I got back, I was tested. I was Lyme positive. And it's a well-controlled experiment because I had given blood before I left. I was negative, so I seroconverted during that time. The point is, I could tell when I first got Lyme from a simple smartwatch and a pulse ox, and then we looked at the data retrospectively. I'd been ill 4 times. 1 was Lyme, 2 were viral infections, and a 4th I was asymptomatic, but I had high CRP, so I knew something was off. And every single time, my smartwatch showed a high resting heart rate and skin temperature in advance of symptoms. So we went on to write algorithms that would actually detect this, and they worked on me, they worked on others in our cohort, and then along came COVID. If you think about how we measure COVID and illness these days, we use temperature, which is a 300-year-old concept, and it doesn't work very well for COVID, right? Half the people don't get fever. I didn't when I got COVID. And so, in fact, actually these days you use these devices. They're high tech, but if they measure you when you're outside, I always measure 33 and they wave me in anyway, even though it's a worthless measurement. And so we think there should be better measurements for trying to follow this. So obviously you can use antigen PCR tests, but you're not going to do those continuously. So we think the wearables could be quite powerful for this. So when COVID hit, we partnered with Fitbit, had 5,300 people enrolled. 32 of them actually had COVID while wearing their Fitbit and had a diagnosis day and a symptom day. And so this is our very first case. Day 48 is a diagnosis day. That person was diagnosis positive then. Day 47, their symptoms first appeared. But if you look at the resting heart rate, it jumped up 9.5 days prior to symptom onset. Very clear signal. Okay? And in the end of the 32, for 80% of the people, we could tell when they were getting symptoms getting COVID, they got to jump up a resting heart rate 4 days median prior to symptom onset. So 80% of the time it worked, 20% we have a hard time getting a stable baseline, but it works for most people. And as I say, the median is 4 days prior to symptom onset. The bottom is a real-time detection system we've set up where we actually follow people's baseline in a circadian fashion and look for an elevation in resting heart rate and other parameters, mostly resting heart rates, most of the score, in terms of magnitude and duration, and then it sends a red alert. So we built the system. It's a cloud-based system capable of handling millions of people. We have, I think, about 10 or 20,000 people enrolled now. It works about 80% of the time, same number. Right now, you still have to click on the app. Soon, I hope we will send you a red alert without you clicking. But basically, this is one of our first cases again. This is an individual who basically had symptoms showing on that day 21, diagnosed the next day at day 22, but actually had red alerts for 3 days prior to symptom onset. It works for Apple, it works for Fitbit. Please sign up because we're trying to make our algorithms better. I'm not sure, I guess the website's somewhere else, but it'll be on one of the slides. It works even in asymptomatic cases, same number, 80% of the time. Here's a case here where this asymptomatic individual was diagnosed on that purple day there. They were getting red alerts for 2 weeks prior to symptom onset. It's more sensitive than your antigen test. We have examples both from me and others where you'll actually get a red alert prior to being positive on an antigen test. I won't show you that. So we think it's, again, quite sensitive. It's not specific for COVID. It's really an anomaly detection system. And if we look at COVID-negative individuals, the number one trigger of red alerts is workplace stress. Okay, so we think, bottom line is it finds many kinds of abnormal signals. Many of these you can contextualize. Other infections will trigger these red alerts too. I'll just say you can measure more things with a smartwatch than simple infections. You can measure red blood cell count, other things as well, using machine learning algorithms. I'm going to skip over this. We do a lot with continuous glucose monitoring and people's specific responses to food. I want to spend a little time on this one. We've been working hard on this idea about doing remote monitoring, where you collect samples at home and mail them in. And after spending about 7 years, we've settled on several devices that work pretty well for stable analytes. We collect blood in fixed volumes, FedEx them to us, and then we do these deep omics measurements. We measure about 2,200 molecules out of a single drop of blood, about 20 microliters. And we can follow then how most analytes are stable, some aren't, we know which ones aren't. And then we do fun experiments. We had 32 people drink this Ensure shake, and it turns out everybody responded differently. So hundreds of molecules change. We can measure glucose-important molecules like insulin, incretin, which are very hot these days, inflammatory molecules. And again, we're sampling them before, 30, 60, 120 minutes after drinking the shake. Here's the cool result. As I say, everybody responds differently to the exact same shake. So each box is a different person. S13, those persons' carbs plummet after drinking the shake. S21, 3 over, goes way up. You can see over, many people's go up, some go down. It just varies with one person to the next. They behave similarly with the class of molecules. We can group people into 5 classes. The cool result is down here, so I guess I will move over here. Gray is the average. These are inflammatory markers. So in this class of people, the inflammatory markers go down when they drink the shake. In this group of people, they go up. Same with this group of people, up. Same shake can be pro-inflammatory in some, anti-inflammatory in the others. 10% of people have inflammatory bowel syndrome. We think this is a way to measure this and try and figure out the causes. Again, we all respond differently. If I showed you the glucose one, you'd see the same result. Here's the ultimate experiment. We actually had one person give samples every hour for 7 straight days during waking hours. Thousands of molecules change. They're wearing a continuous glucose monitor, smartwatch, and food logging. And then we do associations between activity, physiology, biochemistry, and see what kinds of things we can learn and tease out potentially using time-lag correlation, causative events. Doesn't prove causation, but nonetheless, you can get hypotheses here. And what we find, again, thousands of associations between, say, heart rate steps and glucose with all kinds of biochemistry, including cytokines. The cool result is the top and bottom. Let's start with the middle, though. That's actually the insulin C peptide, goes up 10 minutes after this person's glucose. kind of thing you expect, but you can actually measure it at an individual level. We can find new inflammatory markers go up in response to glucose. That's the one at the bottom. And the cool results at the top, alpha-synuclein involved in dementia and Parkinson's, actually has a very interesting pattern. We think it's associated with specific stresses. We're trying to further understand that with the idea that if you can actually measure this, you might be able to mitigate people's, if you will, alpha-synuclein peaking so that you can try and push off dementia. That's the hypothesis anyway. So that's something we'll explore further. I can tell you we're bringing all kinds of wearable omics data into dashboards to try and better have people individually follow their health. How do we get this out to the world? Well, I'm in Silicon Valley, so we form companies, as you heard already. And so we spun off many companies around this. QBio does deep data, actionable deep data, though, not just data for data set. Has to actually turn in something people can use. And I think that's the key for this. It's easy to do research, which I think is fun, and everything I showed you before is research. But actually to translate it, you have to have actionable findings. So we spin off these various companies. January AI is a CGM company. YOLO might interest this crowd. It's a metabolic profiling company. They're generally profiling people quarterly for about 500 metabolites in a semi-quantitative fashion. These things span 20, if you will, health and wellness categories, all kinds of areas. I've just listed 8 up there. And we can drive aging markers. And we have combinatorial scores that contribute to that. So you can see the subcomponents of the aging that are contributing. This is very much like the ageotype I was telling you earlier, the aging patterns that will actually contribute to your biological age. So anyway, this is our way to try to get this out to the broader community. And then actually people do act on some of the information. They can shift their lives from lifestyle changes, shift their metabolic profiles. There's obvious recommendations you can make based on food and other things to try and alter these lifestyles. So this is how we're going to try and get, at least from the metabolic side, out to the broader community. So I'll wrap up and say we think big data and longitudinal profiling can be very powerful for keeping wellness going, if you will, extending health span. This is my wearables team when it was at its peak, microsampling team at the bottom, and Xiaotel Shen did all the nonlinear analysis on the omics profiling. So probably race through that pretty fast, but I'm happy to take questions if we have time for any. But I may have used a little bit too much. We'll find out in a minute. Thanks. There's one way over there and one up front. Well, his was up first, so we'll go to the back.
The heart rate monitoring and finding infections that much earlier than symptoms onset seems really exciting. I'm curious if you're willing to speculate at all as to why that's happening, and also, do you think it's likely we'll be able to distinguish between things like bacterial and viral infections, or even what kind of infection you're getting? Because, our antivirals are oftentimes not very effective if you take them when symptoms have already happened. So.
Yeah, so why is the heart rate going up? Well, I do think your immune system is getting mobilized. We do see changes, and so I think your energy patterns shift pretty quickly. I think that's leading to this increase. It may lead to slight increases in temperature that we just don't pick up by other means. So I think it's a metabolic shift, and probably strongly in your immune system that's causing the increase in heart rate. Let's see, the second question, sorry, what was that again? Oh, yeah, bacteria versus viral. That one I'm pretty sure of. You can look at the signature between Lyme, which is a form of bacteria, spirochete, versus viral infections, and they're very, very different. Now, I think in part, respiratory viruses obviously clobber your lungs. Respiratory bacterial infections, I think they'll still be different enough that we'll probably tell them apart. Whether we'll ever be able to tell the difference between influenza and COVID, I don't know. That one's a tough one. So I'm not so sure about one virus from another, but I think bacteria, like the mycobacterium infection we picked up, also gives a very, very different signature than the respiratory viral one. So I'm pretty confident. This is why we want more people in the study, collect more data. I know, for example, I'm sure we can tell the difference between workplace stress and a viral infection if we have the right data types. It's hard to get the data, actually, the data you want from companies and such. We're probably out of time then, is that right? Yeah, all right, thanks again.